Hi students, this is Mr. Eckert. We're going to go through target C16, which says that you should be able to describe the steps in protein synthesis and differentiate between transcription and translation. And I do want to say at the start of this target that students who are successful, this is probably one of the more complex targets that you're going to encounter in biology, but students who are successful definitely spend time reviewing this target multiple times, if not every day. And you definitely want to try to get the big picture, not just memorize steps in this target, but try to understand the big picture of what's going on. And what will help with that is if you look at some of the help links that I put in the help folder in this set of targets. So there's video clips in there you can watch, there's tutorials you can go through, and taking that extra step is really going to help you understand this complex target. So first of all, we may have talked about this in class already, but every cell faces a fundamental problem when they try to make proteins, because the instructions for making every single one of the 2 million different proteins that the cell needs to make in a human is found, of course, on the DNA, which is locked up in the nucleus. But the location for making those proteins is, of course, the ribosomes that are floating around in the cytoplasm, the free ribosomes, and those that are on the endoplasmic reticulum, as you can see here. So it's a location problem. We've got the instructions in one place and where we're going to make the proteins in another place. So how does the cell solve that problem? Well, the first part of that solution is a process that's called transcription. And it starts out, this is going to sound very much like DNA replication to you, I'm sure. An enzyme called RNA polymerase attaches to the DNA at the beginning of the gene. Now, a gene is just simply a segment of DNA that codes for one particular protein. It's a set of instructions for how to make one protein. So here in this diagram, this thing that looks like an egg, that's the RNA polymerase enzyme. So it's attached to the DNA, and it's going to unzip the double helix at the point of that gene and begin to read the instructions, and those are coded, of course, in the sequence of those A's and T's and C's and G's that you see here. So here's the gene that it's reading. As it reads those letters, it's going to copy it by attaching complementary RNA nucleotides. And the RNA nucleotides bond just the same way as the DNA nucleotides do. C to G, G to C, T bonds with A. Now the only thing is that RNA doesn't have any T. So when it encounters an A, when it reads an A on the DNA, instead of bonding a T, it's going to bond a U, which stands for uracil. So when it's done reading that gene, what results is a strand of messenger RNA. It's called messenger RNA because it's carrying the message or the instructions for how to make one particular protein. So of course, if we're making a copy of that gene that's on the DNA is found in the nucleus, so that is where transcription takes place. The DNA can't leave the nucleus for a couple of reasons. One, it's just simply too big, being a double-stranded molecule, to get out through the nuclear pores. And it's not very safe. It's protected inside the nucleus. If it leaves the nucleus, it's open to the possibility of getting damaged somehow, or maybe a virus could attack it. And that's simply, it's too risky for it to leave the nucleus. So once that mRNA strand has been completed, and again, what it has done is copied the message for how to make one particular protein, it is going to leave the nucleus. It's only a single strand. It's very short, as opposed to the DNA, which is double-stranded and very large. So it's going to go out of a nuclear pore and take that information out into the cytoplasm. Now, of course, once it gets out there, it's going to find the organelle that makes proteins, and that is the ribosomes. And so the second part of protein synthesis, making a protein, is called translation. So first we did transcription, that's inside the nucleus. Now out in the cytoplasm, we're going to do translation. So it's going to take the language of nucleic acids on the messenger RNA. It is recorded in those actual those nitrogen bases, the A's and U's and C's and G's. Again, we're not using T this time because we have RNA instead of DNA. And so it's going to change it, it says, into the language of amino acids. Well, what it means by that is every protein is made up of 20 different combinations of 20 different amino acids. So it's taking the A's and U's, C's and G's and changing those into the ingredients for how to make the proteins, and that's the amino acid. So it needs a translator in order to do that. The translator is the ribosome. So the ribosome is going to hook up with a messenger RNA molecule, and it's going to start to read it. It reads it 3 letters at a time, and it starts at the letters, every protein starts with the letters A, U, G. That is known as the start codon. So every sequence of 3 letters on messenger RNA is called a codon, and the sequence AUG, that unique sequence, is known as the start codon. So that kind of lets the messenger, or I'm sorry, that lets the ribosome know that once it reads those 3 letters, AUG, what follows that is going to be the exact instructions for how to make the particular protein. And that it reads that in what's called the P site. There's 3 different locations inside of the ribosome. The P site is where those 3-letter codons get read. So the ribosome reads that code, and that tells it what amino acid. So that every 3 letters, those codons correspond to one particular amino acid that it's going to need as an ingredient that goes into making the particular protein. Now, how those amino acids get to the ribosome, they're brought by these molecules here. Kind of reminds me of a pop can that's kind of knocked over on the top half. But this molecule is called a tRNA. The T stands for transfer, because they're transferring these amino acids. Here it's the amino acid methionine. Methionine is always the amino acid that corresponds to the codon AUG. So the tRNAs bring those down and bond. You can see on the end of the tRNA there's 3 letters as well, and they're complementary to the codon. So they call these anticodons. So on the tRNA, on one end is an anticodon. You can see here it's a UAC. On the opposite end is an amino acid. So they come and temporarily bond to the messenger RNA in the ribosome. So then another tRNA molecule comes down and attaches at what's called the A site. And it is bringing a different amino acid, or it could be a like amino acid, the same amino acid as the first one. But it's bringing another amino acid. Those 2 amino acids will bond together by what's called a peptide bond. And then the tRNA that was originally in this first P site here, it moves over into the E site, and you notice that it detaches. And notice this one here does not have its amino acid anymore. That's because it's bonded to this growing chain. So this process is going to continue to happen. This messenger RNA is going to move through the ribosome kind of like a ticker tape. It's reading it 3 letters at a time, and once the tRNA comes down, leaves its amino acid, it's going to move forward, the mRNA. It's going to read the next 3 set of letters. A new tRNA molecule is going to come down, bring its amino acid. The amino acids bond together with peptide bonds, and it keeps on doing that until it reads on the messenger RNA what's called a stop codon, and that tells the ribosome to do just that, to stop. You're finished making the protein at that point. So once the ribosome reaches that stop codon, it says everything detaches, and by everything we mean this growing chain of amino acids. That's what forms the protein. So it's going to detach. All proteins have a certain shape, so the protein is going to be folded, this long chain of amino acids, which is also sometimes called a polypeptide, because remember, the amino acids are held together with peptide bonds, so poly means many, so that's where you get the term polypeptide. So it folds into a particular shape, and that determines the function of that particular protein. The messenger RNA then gets recycled by digestive enzymes. You may remember that those are in lysosomes, And so then the parts of the messenger RNA will be recycled and go back into the nucleus. And each tRNA, transfer RNA, that dropped off an amino acid, it'll go back out into the cytoplasm, pick up another of the exact same kind of amino acid so that it can be used again to help build another protein. So I would encourage you to watch. We're not going to take time during this video to watch this during this PowerPoint to watch this video clip, but this is the one that we've watched in class. If you type in replication and transcription and translation on YouTube, you'll see this video here.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular.
That particular video clip is about 6 or 7 minutes long, so if you want to look that up, I would encourage you to watch that, and watching it multiple times will actually help you to get this process down. So that's it for Target C14. If you have any questions, please make sure you ask. And again, I can't encourage you enough to use the help folder, look at those additional video clips and tutorials to help you better understand that target.